The most common sentence of people finding new organisms that are interesting is something like, I read about this organism and it looks amazing. But then I Googled it and there was absolutely no publication, no lab is working on it. So I dropped, because no one wants to do that. No one wants to take the 10 years that it takes to just figure out how to get a plasmid in, figure out what it grows in, figure out if it's aggregating in a tube. And so I think success for us would be that people manage to actually adopt more organisms.
Hey, Ram, welcome home.
Hi, Karl. It's great to be back. My brain's exploding.
Yeah, so for those of you who haven't listened, we just got back from Simba Beta, which was held this year in Oakland for the second year. It was your first time at the premier synthetic biology conference. What did you think?
Like I said at the top of the episode, my brain is exploding. So much is going on in biology and biotech. Everything from healthcare to biomaterials to cultured meat to space. I didn't even think about that at all until I got there. And now I'm a big space biomanufacturing evangelist.
That's awesome. There were 3 action-packed days of panels and keynotes, followed by some kind of networking event or event at a bar or a dinner. And there were multiple tracks running. There was a track on decentralized science. There was a track on the tools of biotechnology. I can't even keep track of the number of tracks that there were, and I could not even get to many of the panels because they were so full. But I think for me, the main thing was just I probably spent 70% of my time talking to people, seeing friends, meeting new people, and then 30% in panels. Erum, so you said space. Was that the biggest surprise for you? What was your biggest surprise?
Yeah, space was really interesting because there's so many disciplines that are coming together. First of all, the premise is to think about going to space and being able to terraform different places in space, whether it's a planet or creating like a spaceship. Earth is a spaceship, as our friend Eben Bayer always says, spaceship Earth. Can we recreate a spaceship where we just live on that off of Earth? And the premise is that Earth is a finite resource. The population is going to explode if things go well. So considering living in other planets is a good idea, perhaps. So then what does it mean to have biology in outer space? How do you think about making drugs in zero gravity or microgravity or differential gravity, as I learned? And what is the biophysics of that? And how can that lead to new gene expression? I thought that it was so fascinating, and they go on that. I think what we need to do for our podcast is get Kate, her last name is escaping me, but she is an astronaut who is a biologist and who's working on these challenges in space at the space station. We are going to Moon. We are going to Mars. That is definitely happening as part of NASA's missions. So we're going to be hearing a lot about what experiments they're planning on doing as we go outer space.
It
so
still seems
super fascinating.
Yeah, it still seems a little unreal having grown up through the Apollo missions and then the space station, the ISS. But now it definitely seems like we're going to space pretty soon, and it's going to become a part of our everyday lives. The other thing that we heard a lot about was just the impact of biotech on climate. There were a couple of things that really surprised me. One was how quickly things are moving. The things that used to take a postdoc 4 years or 5 years can now be done by an undergraduate or even a high school student in a matter of months. The easiest example would probably be sequencing a microorganism. That one is something that high school students can do, and we're going to talk about that on the podcast. But things are getting much easier for people to do with biotechnology. The other thing, besides the number of people that were there, it was almost 3,000 people, was some of the revenue numbers that we heard from consumer biotech companies, companies that are actually making products that are consumer facing. We have featured Charles Dimler of Checkerspot on this podcast. Their company makes skis, is getting into apparel. Lanzatech, who we haven't had on the podcast, but we hope will be on here, takes CO2 emissions and turn them into a thread. So seeing those products was incredible.
Lanzatech was really interesting because the threads that they're making, they're already commercialized. They have a partnership with Adidas and they're already making jackets. So we saw our friend Michael Kopke at Beta, and he was wearing this awesome Adidas jacket made from the threads that were made from CO2 emissions that would otherwise have been in the atmosphere. That's just mind-boggling. And the fact that you can do that, I think for us, as we hear about it all the time, I don't want to say we take it for granted, but we're like, oh, okay, obviously it can happen. But for people that listen to this podcast, this is a real thing. Instead of carbon emissions destroying our atmosphere, we can make really cool clothing out of it. It's just mind-boggling.
We should leave a link to that in the show notes because I did look it up on the Adidas website. It's a really cool jacket. It's a really cool shirt. It doesn't say LanzaTech on it, but Adidas does explain that the thread was made by capturing carbon dioxide, which is incredible.
They have multiple partnerships with different brands. And one of the brands does say LanzaTech on the tag. Forget which one, but
I think that's H&M.
I think it's $100 for the jacket, which I'm surprised about. It's more affordable compared to some other biomaterial threads that are turned into clothing. I saw a sweatshirt that was like $500, but $100 for this jacket that LanzaTech partnered with Adidas on is phenomenal. So we'll put that in the show notes if you want to order it.
Absolutely. So then what, of the things that you thought you were going to see, was there something that you were surprised wasn't being talked about?
They had some incredible speakers, some heavy hitters in the biology space, as well as the mycology space, as well as television. So we had Craig Ventnor, the father of sequencing the human genome, talk about xenotransplantation, which is taking organs from other animals and using them to replace organs in humans. So if you have a faulty heart, perhaps a pig heart or a heart from another animal could suffice. He spoke with Martine, what was her last name?
Rosenblatt. She's the founder of United Therapeutics.
Yes. And she was on stage because Craig is on their board of advisors. And she said repeatedly that their mission is to create an unlimited amount of organs for everyone on the planet. They have a very strong vision of making sure that our organs don't fail us, and that won't be the reason that people die anymore. And it's just amazing to hear that story because Martine was a computer scientist working at Sirius XM.
Yeah, she was the founder of Sirius FM.
Yep. But she had a daughter who had a rare disorder who needed a transplant, essentially a lung transplant, but that was not available 30 years ago. She borrowed from Ray Kurzweil the idea of creating a bridge. So yes, the possibility of having different organs and doing transplants is possible, but we're not commercially there, but we can develop therapeutics to bridge that gap. So she worked on therapeutics for saving her daughter's life, who is now still alive at 39 years old, which is my age. So that was fascinating. It's also very fascinating that Craig Ventnor did say on stage that he believes that COVID was a lab leak.
Interesting.
And for someone that has that much prestige and scientific rigor to say that does bring up some questions and it's making me rethink. It happened. We should now take a look and investigate. And I just wonder what the results of that investigation will be. No one should get fanatical about it. I know that's the way some of our population works, but it's just interesting that he said that on stage. And then another heavy hitter, of course, one of my favorites was Paul Stamets. The guy is amazing. If you guys don't know Paul Stamets, you might know him from the documentary Fantastic Fungi on Netflix. And he was basically on stage just singing the praises of shrooming. Psilocybin, which I always mispronounce, is very fascinating, but he was really talking about it more from a therapeutic standpoint for people that have depression and post-traumatic stress disorder, or just that are very depressed or confused and maybe just need a new perspective. And this enables that. And he had a lot of research that he presented on stage and a lot of his efforts. And we'll put some of that in the show notes. There's a company called Mycomedica, and the purpose of that company is to create therapeutics using mushrooms. So
yeah,
that was another big thing.
Yeah, I thought he was fascinating. And I'll just add a couple of notes from his talk. One is that these psychedelic mushrooms, which contain psilocybin, are found on every continent on the planet. And the native peoples on every continent on the planet have had a tradition of using these plants as sacred medicine, so for time immemorial. And the thing that really struck me when he was talking about microdosing or even macrodosing was around neuroplasticity and neuron regenesis. So for people who have had, you mentioned PTSD or deep depression, but for people who have issues with pain, it just seems like there's a lot of benefits that are still being explored. It was interesting because I was listening to another podcast and they were talking about being at a dinner in Silicon Valley, and half the people were talking about microdosing, and those conversations were divided into about three. People talking about their experiences, people talking about the science, and then people talking about the legalities of it. So very interesting space to be exploring. Paul was a champion. I really enjoyed that conversation. I love the documentary. I know you got to speak with him. My hope is that we get him here on the podcast sometime in the future.
We'll give him a shout and see if he could come on and talk to our audience about the amazing things that are going on in mycology. And again, we'll put more resources about all his efforts in our show notes because it is very fascinating.
Anything else? You said you had a long list, so I'm letting you go through the list.
I do have a long list. There was a lot in healthcare, just in terms of what's going on in the biopharma space. It was really fascinating because you've been to most of the Synbio betas and they didn't have healthcare, or at least pharmaceuticals as part of their tracks.
There's always been a good presence, a good number of biotech, or let's just say biopharma companies that are focused on drug development or diagnostics as part of the community. Having a track specifically for them was something that was a bit different. I think that most companies, if you're doing some kind of therapeutic in the biotechnology space, either you're using synthetic biology tools or you're calling them something else, but they are a big part of synthetic biology. The other side of it is on the biomanufacturing side, because if you're producing any kind of protein, which is what the best-selling therapeutics are right now, you do need to be aware of what's going on in the biomanufacturing space, and that is all related to synthetic biology. But yeah, there was a number of companies and there was a whole track.
Yeah, I think a big thing that you just pointed out and Paul Stamets pointed out, and you and I have been talking about a lot, is the term that we're using, the terminology to really talk about biotechnology. Like we've separated it into synthetic biology. And I think for people that are in our audience that are not in the biotech space, when they hear synthetic biology, it does sound fake. Like synthetic, means fake or man-made or something along those lines. And Paul Stamets and I talked about that. And he then talked with John Cumbers about it. And Paul was saying that maybe just instead of calling it Synbio beta, just call it bio beta or new bio. I know that our friend Shara from C16 uses the term new nature, because if we're going to be using this and people want to learn more about it, or they start to question like, okay, where did this meat come from? Oh, synthetic biology. And they just make all these assumptions rather than looking into it, which oftentimes people do, that maybe they'll start getting up in arms about their assumptions, which happens all the time. So I think it's something we'll be talking about throughout all of our podcast episodes, is how do we talk about biology? It should just be called biotech in general. And then people are saying that there's industrial biotech, which is what we talk about a lot. And we're talking about personal care, ingredients, biomaterials, textiles, and all of those things. That is becoming a little bit more of a common phrase. It's industrial biotech or sustainable biotech. And then we separated which biotech had been encompassing of pharmaceuticals, but now there's biopharma to separate that out. That was something that I thought was very interesting, as the community of founders and innovators and investors came together. And that was a topic that was talked about, especially us as people that are science communicators, thinking of synthetic biology in a different way, or just it's biology or it's biotechnology. Let's move on.
Yeah, someone said it. Biotech or synthetic biology has a branding problem, and I've never been happy with the term synthetic biology. And then there's engineered biology, which is really biotechnology. And then I don't know if you've heard these terms: white biotech, blue biotech, green biotech. They refer to different kinds of biotechnology. Blue biotech might be something that's related to the ocean. Green biotech might be something that's like more agriculture or forests or land. White biotech might be, and I'm getting the definitions wrong, so we'll have to look them up to clarify. But it's something interesting to think about when we look back at this in 30 years, where biotech is really responsible for most manufacturing. We'll look back at it and just be like, it was funny how we used to call it synthetic biology, or we used to call it biotech. It just is technology applied to biology, and it makes everything.
Yeah, I'll just say one thing along those lines. The conversation I was in, we were talking about the concept of genetically modified organisms and GMOs. I know that that's always been a very conversational topic, but since we were with people that are in this industry, they look at it very differently. They're not evil scientists that are in the lab just trying to take over the world. They're not that at all, but they are thinkers. And consider this: we have been modifying dogs for centuries. That is a form of engineering because it's the technology we're using is breeding. And so they have been genetically modified through human selection. Same with domesticating cows and sheep, same with plants, everything. Although it's not using very complex technology that is not well understood, which then people are like, I don't understand it. It's evil. If you don't like genetically modified vegetables and fruits, then you don't like your dog. That was something,
yeah,
or your cat. So that was something that a new perspective that was introduced to me. And I thought, oh, that's very interesting because sure, there's engineering and using CRISPR and all that. And that's something we do because now we have more precise tools to do it. But we have been doing it with not precise tools, with just guessing games of selective breeding. And we've been doing that for years or centuries.
Yes.
So I thought that was good perspective.
Yeah, I think that perspective is always worth making and to remind people this is nothing new. It's just the tools are new and our ability to do it faster is new. Biology still is what it is. You can't necessarily brute force it to do things that it wasn't made to do. You can try. It doesn't always succeed. And if there is nothing else that you want to cover on SymBioBeta, because we can go on and on about it, let's talk about our interview. Speaking of brute forcing nature.
Yeah, I'm really excited about talking to Nili from Cultivarium. It's something that we have become intimately knowledgeable about because we do work with Cultivarium on a day-to-day basis. They are working with non-model organisms. And so what does that mean? Nili will get into it, but ultimately a lot of our biotechnology is built on a few we call workhorses, a few microorganisms that we do brute force engineer, like E. coli, S. cerevisiae, which is yeast. So when she says cerevisiae, it's yeast. Nili and her co-founder Henry studied a non-model organism, which took them years to what we call domesticate, right? Domesticating cows. We domesticate them to make milk because that's what they're good at. But we're trying to do the equivalent with E. coli, a microbe found in our guts, to do things that are meant for the earth. So it's like we're really brute forcing it, but why not use different organisms?
Okay, so I'm excited about this podcast. I think we should warn the audience that it does get a little technical, and we're going to do our best to help you understand. But just to set it up, the biotechnology industry is basically a $1 trillion industry in the United States. And as Erum mentioned, and as I mentioned, it's built on just a handful of microbes. The most famous ones are E. coli, the gut microbe that we all carry with us, and brewer's yeast. And we do a lot of really interesting things with them, but we try to make them do things that they're not designed by nature to do. And there's 1 trillion species of microorganisms on the planet. If you take a tablespoon of soil, it contains more microorganisms than there are people on the planet. So if we were able to work with those organisms and understand them better, and we can understand them faster, the thesis that Cultivarium has is that we would suddenly expand biotechnology exponentially. Nature has given us a whole toolkit that we have yet to access. So I'm excited about this. Now, I think it's worth it to set up a couple of definitions in advance because there's going to be a few throughout. Erum, do you want to take a few of them and then I'll take a couple of them?
Yeah, sure. Like I mentioned, Henry and Nili, they worked on a non-model organism called Vibrio nitrogens, which is a halophile. And a halophile is halo is salt, phile is loving. They're salt-loving microbes. And as you can imagine, salt-loving microbes would be thriving in oceans, which have high salinity and make up most of our planet. So when she talks about Vibrio and halophiles, now you guys know what she's talking about.
She also mentions NGS a number of times, which is an abbreviation for next generation sequencing. Erum and I were talking about how biotech has gotten so simple and is moving so much faster. A lot of it is thanks to the technologies of NGS, next generation sequencing, which make it much easier to sequence an organism. A human genome is very large. It's something like 3 billion base pairs, which is the number of chemicals that make up your DNA. But a microbe could be very small, and some are very small. So using these next generation sequencing tools, you can sequence some of these organisms in a couple of days, maybe even overnight. So she does talk about that. Then she also talks about plasmids, and plasmids are circular pieces of DNA that exist naturally. It's one of the ways that bacteria communicate with each other. And in biotech, we use plasmids, circular pieces of DNA, to transfer information from one organism to another, to figure out how an organism works, to turn off certain genes. So that's something that Nili is going to talk about, just to give you the background on that.
The plasmid is where you can also engineer the DNA, correct?
Yes. You can put DNA into a plasmid that then you drop into an organism. And if you do your work the right way, it becomes a part of the organism's DNA sequence or genome, and then it'll produce the protein or the behavior that you are looking to produce in that organism.
And Nili will point out that is one of the most challenging parts of working with non-model organisms is getting the DNA delivery, getting the DNA into the microorganism or bug. So she uses that term interchangeably. So when she says bug, it's not like a tick or an ant. It is a microbe, just to keep that clear.
So let's get Nili on, and then we'll come back and talk about the interview.
I am so happy to start this interview with Nili Ostrov, one of the co-founders of Cultivarium, a good friend of Messaging Lab.
She's one of my favorite chief scientific officers. I have a posse of many chief scientific officers now. Sorry, you're not the only one, but you are one of my favorites.
Happy to be in your posse.
Yeah, you're my
I'm
posse.
honored.
The CSO posse. We'll have to have a gang sign. But yeah, we want to know what sparked your interest in biology? What got you down this path to where you are now?
I think that the first earliest thing is that I grew up in a household where my dad had all these animals brought in. We had rabbits, we had birds, we had parrots. I believe my dad actually had snakes before he married my mom, and she vetoed the snake habit. But he really loved just taking care of animals. And I think from a very young age, I loved doing that and had interest in that. As I officially started to learn biology, it just opened a whole world of how things work. And I had great teachers, and I think it just sparked this interest of finding how things work and seeing what's underneath the beauty of nature. And initially, I actually wanted to do zoology and go out to the jungle. But then I realized you actually need to go out to the jungle, and you actually need to open up an animal to see what's inside. That's the least favorite part for me. So I think microbiology is where I find myself most comfortable. It's fascinating, and it's molecular biology is the part where I really find the most interest.
Let's talk about that for a second, Nili. If someone doesn't know what microbiology or molecular biology is, what are those? How do you tell people what those are?
Microbiology has been there forever, right? Like human history is basically paired with microbiology because we make food with microbes. And so there are microbes everywhere all the time. We're just not as aware of them. And then molecular biology really took a central stage in the last few decades where we started understanding and having really good tools to poke at the inner side of cells and understand how things work. And with DNA technologies and PCR and the really advanced tools for us to be able to edit cells, it changed everything about biology. It's no longer the observation that matters. It's really how things work. And so the observation is paired with how does this thing really happen? How do these colors come about? What does this little motion really mean? How do ions move around? It's just a very interesting way of understanding how we function and how the world around us functions. So molecular biology is unraveling the secrets of the insides of the cells.
So clearly you're very knowledgeable in microbiology, chemistry. You have a PhD in chemistry. You've been living and breathing the sciences for years. You know what's going on in the space. And you and your co-founder decided to create a company, or actually it's a focused research organization, which I know you're going to get into, and it's called Cultivarium. So what is Cultivarium and why are you doing this?
So the very simple answer is Cultivarium is a startup, and Cultivarium is trying to use non-model organisms to advance biotechnology. So if you step back and think of all of microbiology and molecular biology and all those fields that are very well-established biological sciences, most of them really just focus on a couple of organisms, which is funny, right? We're surrounded with trillions of different microbes around us and in the world. But really, most of what we know is based on just a few organisms. E. coli, cerevisia, right? The yeast, Saccharomyces cerevisia, mice, a nematode. And so there's a few selected organisms that everyone uses for everything. And Cultivarium was really started to try to break that barrier. And that barrier is a fairly high barrier. The reason we started Cultivarium, I met Henry, my co-founder, when I joined George Church's lab for a postdoc at Harvard. And that's a place where a lot of people do a lot of really crazy things and try to push technology and push biology to its limits. And Henry was doing the unthinkable. He was choosing a non-model organism to work on. So everyone worked on E. coli and human cells and yeast. And then this one guy is choosing a Vibrio from some salt marsh and tries to figure it out. So when I just joined, there were a few months where you just meet everyone and talk to everyone about their projects. It was really interesting how the most crazy thing you could do, not the most crazy, the lab had a lot of crazy stuff, but the most out there thing you could do is choose a non-E. coli microbe to use for technology. And that says something. If we used to have whole PhDs just to try to sequence 1G years ago, now you just order a sequence. But you still need a whole PhD or a whole postdoc to get a non-model microbe to work. That tells you how much that field of non-model organisms is really open and really is a place for exploration. So the way I got to do Cultivarium and start this with Henry, my co-founder, is that we spent our postdoc trying to figure out how to work with a really fast-growing organism. It doubles half the time of E. coli. So it doubles every 10 minutes. It is really fast. And you could tell just intuitively that's super useful. If you could do anything you do with E. coli half the time would double the yield, that would be amazing. So it still took us a very long time to get basic things, to get sequences the fastest, right? But it's really difficult to find how to grow things, how to engineer them, how to manipulate a new bug. And so that process cannot take that long. It's impossible for us to work with the trillions of organisms around us if we take 10 years to figure out how to get a plasmid into one organism. So Cultivarium is about lowering the time and cost that it takes to onboard new microbes. That's why we're here. And we're going to try to do that in weird microbes, and we're going to try to let people have tools. We are tool-focused, technology-focused. Let people do that themselves. Open the floodgates by allowing people to do it themselves.
If you are successful, what does the world look like based on Cultivarium success?
The first thing that I would really love to see is the bioeconomy being based on multiple organisms. Right now, most things being fermented for actual products. So we're very early R&D, clearly. This is organisms that people don't know how to work with. But if you look at the other end, where drugs are made, where products are made, where food alternatives are made, most of these scale-up production processes are based on a few organisms that we already know how to work with. But there are a lot of other organisms out there. And so I think success for us would be that more people work on more organisms, and using our tools, if that helps, that would be great. I think the more users we can have in terms of both talking to us, using our tools, doing it themselves, using our computational tools to just find out about organisms they've never learned about before. The most common sentence of people finding new organisms that are interesting is something like, I read about this organism and it looks amazing. But then I Googled it and there was absolutely no publication, no lab is working on it. So I dropped, because no one wants to do that. No one wants to take the 10 years that it takes to just figure out how to get a plasmid in, figure out what it grows in, figure out if it's aggregating in a tube. And so I think success for us would be that people manage to actually adopt more organisms.
Because of the structure of your startup, which I would love for you to dig into a little bit, it will make it easier for people to leverage all of the databases, protocols, tools that you guys are creating, because you're not charging for it.
Yeah, so Cultivarium is currently fully philanthropically funded. We are a member of the Schmidt Futures Network. We are going to release our tools and make them available to as much of the scientific community as we possibly can. One of our goals is to reduce the barrier for sharing information, reduce the barriers to approach new bugs and new microbes and find information about them and be able to use them. So we're developing a portal, and the portal, we hope, is going to bring together a one-stop shop for anything you wanted to know about a new microbe that you literally had nowhere else to find. And so things, for example, like if you want to work with a strain of Vibrio you've never heard of, the species name, but what if there's 7 different strains? What if the strain bag that you look up this name has 8 different strains from 8 different environmental niches and you have no idea what the difference is? And so we bring together all of these pieces of information. The other thing we're doing is to actually do work. We have a wet lab and we generate data that tells you on the portal, how well does it grow in LB? Can I clone something into it? Does it have any special restriction modification sites that are known? What is the sequence? All these basic things that take years to find on your own, We hope that by being able to put all of these things in one place and letting people see that, with the caveats, of course, of biosecurity, we're very heavily considering how to best release this kind of data to make sure that we are in compliance. I hope access to that information will be the key for people to not be afraid and not have the issues that we had when we started with a new organism.
That's amazing. When I think about Cultivarium and getting to know you and Henry, I think, and you can correct me if I'm wrong about this, I think about this mandate of making tools openly available, of course, with the asterisk of biosecurity in front of that. But making them openly available actually is a huge, potentially economic driver. And the analogy I think of is on the internet, so much of the internet was built on open source tools. Most people never even consider the foundation of what is behind this thing that permeates our lives every day. But a lot of it is built on tools that were developed in groups that shared them with the rest of the world, and other people can go in and modify those tools. Is that kind of a good analogy for what you're doing with Cultivarium?
Yeah, I actually think that's a great analogy. I think you really found a connection that speaks to people. With the internet, I think if there's something that you really don't even consider doing, but then it becomes available and you see that it's not that hard and you can literally at the tip of your fingers have information that you previously didn't have, it drives change. So one of the advantages of us starting Cultivarium in this way, where we release tools and we try to engage people and try to interact both with academics who are really at the forefront of using weird organisms that we don't yet know what to do with, and companies that use organisms that are more mature, we can talk to any of those groups and we can drive change in specific organisms, but also we can drive change in the entire community being able to adopt organisms. So I think open source is key to drive engagement. And by doing Cultivarium in this way, we're able to offer tools to more of the scientific community members. And we talk to nonprofits and we talk to for-profits and we talk to academics. And I think each one of them have a different need, and each one of them we can approach in a separate kind of tool set to really offer any of those, something that Cultivarium can do to help with their organism or to help with their tool that they're having issues with. I can't tell you how many people I talk to that tried an organism, spend their entire few years of postdoc or PhD really banging their head against the wall with some delivery system of some plasmid that didn't work. And it never saw the light of day because you usually don't publish negative results. And so our goal is to publish negative results as well, because if something doesn't work, I don't want anyone else to spend the next 5 years trying to do that. So I think the way we are structured helps us provide insight to this community that otherwise we might not be able to.
From a macro point of view, I want our audience to understand the benefits of not working with yeast or not working with E. coli. What would be the benefit of working with a non-model organism once they're available?
Yeah, so there's multiple answers to that. One of them is, I don't know, and that's good. And so one of the amazing things about non-model organisms is that there are so many phenotypes out there in nature that we can't even imagine and we don't even consider using because we don't know. And so one obvious benefit of using a non-model would be if something grows in the ocean and you are trying to bioremediate oil spill in the ocean, maybe your own gut bacteria that grows at 37 degrees in an anaerobic community in your gut is not the best organism to try to engineer and put out in the ocean to clean up an oil spill. Maybe if you're trying to do agricultural crop science, you don't necessarily want to use Cerevisia that doesn't belong to that community. And so the obvious advantages is choosing organisms that could do functions that we don't currently have in our current model organisms. E. coli is wonderful. It's really great. And Cerevisia is an amazing tool. But I don't know that it's fair for us and for the model system to ask them to clean carbon and then also bioremediate and also make drugs. The organism itself is not geared towards all of these things. And it's amazing that we've been able to do that with so many different applications for just a few organisms. But then imagine what we could do if we only had a few more. What if we had an ocean-based and then a soil-based and an Antarctic-based and a halophile that can all do what E. coli could do, and you could just choose. So I think the I don't know is a really good answer. And that's also why we are explorers, and we're not necessarily starting with the answers. We're trying to open options that are not currently available.
Going to be making a lot of friends in the life science community by doing this work, because they can get to maybe where they're going. If they have a hypothesis to clean an oil spill using another microbe that they have a hypothesis on, and you can provide that information and save them so much time to get to where they're going, it's going to be great for them. But we're also talking about trillions of microbes doing a lot of complex analyses and creating these protocols. Does sound like a very huge undertaking. So how are you organizing your approach and the stages of research that you're doing?
That's a very legitimate question. We started by thinking we could do any organism, mammals, everything, changing giraffes, but very quickly, we are microbiologists and we focus on microbiology. And the way we think about it is there are three major things or achievements you would like to have in any microbe in order for it to be used for any application. You need to be able to grow it in a lab robustly. You need to be able to get DNA in, and you need to be able to engineer it. If you don't have these three things, it is useful, but not as useful for most applications. So most people who use any strain for any application would like to enhance their strain and optimize it eventually. And so natural organisms are wonderful, and some of them are actually super useful, but eventually you want to be able to engineer a strain to open up applications. And so we focus on those three things. We onboard organisms and we ask what's missing. If no one can grow it properly, then the first thing we do is try to figure out how to grow it. And so we look at a variety of medias and we say what makes sense for us to try. And then we, of course, automate and multiplex, the two magic words, to try to get as much information as we can to provide insight into how well does this organism grow in those types of medias. And then we ask, how do you get DNA in? And that's where most people stop, right? Most people that try a new organism can grow it. And we have strain banks with a lot of different strains that people could isolate and grow. It gets hard when you need to change the organism and get DNA in. So some organisms just don't like any of the current methods of getting DNA in, right? We don't know how to conjugate them. We don't know how to electroporate them.
All right, here we go. So Nili mentioned how to conjugate or electroporate new DNA into a cell. Okay, so conjugation is when one bacterium transfers genetic material to another through direct contact. Electroporation is using an electric pulse to temporarily open up cell membrane pores for DNA to get in.
A lot of these methods are a little bit bespoke, right? You have to know how to do it or have someone tell you exactly how to use this organism. And so we're trying to develop ways of quickly screening and quickly achieving a protocol to cover a lot of ground and give you some understanding of what is a protocol that you could use with this organism. So it starts with a yes or no, and then you go into efficiencies. How can you make this better? And so once you get a plasmid in, which is not easy at all, if you think about it, I could go back to that in a second, you want to be able to engineer. And engineering is everything we all love, anywhere from recombinering of oligos to full-on CRISPR system and metabolic engineering and pathway engineering and enzyme engineering. All those things require basic capabilities of modifying a genome. Sometimes it's really a chicken and egg problem. You can't really get a protocol to get a plasmid into a cell if you don't know what plasmid to try, but you also don't know what protocol to try. Because you need a plasmid to try any protocol, but you need a protocol to try any plasmid. So that kind of problems is where people get stuck and usually just don't use the organism. So we have some experience with that, and we really like multiplexing and NGS assays. And so we're trying to tackle that in a way that provides some insight very quickly.
I've seen the graphics, what you're calling an organism readiness level. Could you describe the origins of that idea? Where did it come from? And maybe give our audience insight into why having that kind of idea is important or will be important for people who are looking at these non-model organisms?
Yeah, so the idea of technology readiness levels is how well you can use an organism for actual applications. How well does it work with existing industrial processes? If you look at the R&D process of getting an organism even to start being useful in the lab, there's no good definition to how well can this organism be used. So the way we think about it is there are a few basic things that are required to get an organism into our lab. Sequencing is pretty straightforward. So we all know as a community how to sequence things, even if we can't grow them. We know how to extract DNA and sequence and know that something is there, but we can't grow it. So sequencing is almost like level 0, level 1. We don't need to develop sequencing technologies. They are fairly mature. Culture is not as mature, right? Culture is something you have to try to get, to find out, usually brute force. So the first level we're thinking about is how do you get cultured? Can something be cultured? And then the levels above that are things like, can you get DNA delivery in? Can you get true annotation, functional annotation of your genome? Can you get transcriptomics? Can you engineer your strain? And when you get up these levels, we generally define 6 levels. And we say, when an organism comes in, maybe it is well cultured and has a sequence, but no one can get a plasmid in. So where we come in would be to try to get a plasmid in.
Nili mentioned annotation and transcriptomics. Both of these relate to really understanding how DNA is coding for specific functionality or proteins. Annotation is a process of identifying the location of genes and all of the coding regions in a genome and determining what those genes do. Transcriptomics is a study of all RNA molecules in a cell. RNA transcribes DNA and contains information to make proteins and perform other important functions in a cell. While we're here, Nili will also speak about origin of replication, or ORI for short. And ORI is a particular sequence in a genome at which replication is initiated. And that's key for when we're looking to kick DNA replication into gear for successful growth of microbes, especially after we engineer them or have them grow in a lab or environment that they're not used to.
And so one of our first releases that we're expecting to have, maybe even by the time this interview is released, is a system where we can quickly tell what plasmid works in what organism. So by having a pooled approach where we have a lot of plasmids with a lot of different origins of replication together, you do one conjugation into an unknown bacteria that you don't actually have pre-knowledge of what works. And then you just analyze all the colonies that work, all the colonies that grow. You do an NGS assay and you just look at which plasmid managed to be selected in this organism. And you can tell with a single conjugation what worked. So we have a system where we can provide people with a pool of plasmids that we constructed with all the genetic parts that they could use to build their own plasmids if they want to add parts or change parts. And then the protocol and the NGS analysis assay to get the results they need. So if you have your favorite organism, but you don't have a plasmid for, you can try our materials. So our pooled plasmid that you can get from Agene, or talk to us and then do an NGS assay and see which plasmid grows. And I think just that, if I had that when I started a new organism, is pretty significant.
That's amazing. And just to make a comment so that our audience knows, Nili, you mentioned that sequencing is so simple, so basic. Even high school students are sequencing DNA in their biology classes. And if they aren't, they should be. But I recently read a tweet that said something like, you can sequence an organism faster than you can get that sequence published in any peer-reviewed publication.
Yes, I agree with that. And I would like that to be the case for a lot more than just sequencing.
Yeah. And then you kind of described at what point an organism is deployable or potentially usable in industry or usable in the lab. That would be the highest level of readiness. What does that look like? What are the kinds of things that someone would be able to do with a deployable organism? I guess it's the kinds of things that we already do with yeast and E. coli.
Yeah, I think that if you are considering making a product, any kind of product, using biofermentation, then the organism, the host you are using for that product has to be understood, has to be manipulated in a way that you trust that if something goes wrong, you know how to fix it or you know how to approach fixing it. And so I think deployment looks different in different applications and in different hosts. But you know, when you choose a host and Google it or just look at PubMed for 5 minutes, how hard is it going to be for you to troubleshoot? And yeah, E. coli can be hard to troubleshoot, but you have so much information and so many people to talk to. And so I think it's a lot about community. We would like to have a community where people know that someone out there can help them with their organism. They don't have to call individual labs and ask them about their future publication about something no one's never heard of. There is somewhere where non-model microbes are considered, and they are routinely, daily considered as legit hosts for things, right? And people are developing tools, and there's a community where you can talk about them and learn from other people's mistakes and successes. And so I think that choosing hosts for application should be an easier process. If we actually want a thriving bioeconomy, then we need to use more of the bio. We need to go through our biosphere, our own biosphere, and find more tools that are already there that we don't know about, and try to use them and try to make them systematically easier to use. And I don't think people are not trying to do that. It's just that it is hard to do. And it's only hard to do because the technology is not yet there to make this specific type of host available and easy to use. And so we can put together a lot of the methods and the tools and the protocols that people use for E. coli, and each one of them will need a lot of tweaking for each host that you're going to choose. And you asked me before, what does it mean? There's trillions of organisms out there. How are you going to do this? So the way I think about it is the most useful tools we can make are organism agnostic. And by that, I'm not saying all organisms are the same, not at all. Or each organism has its own special issues and it's a snowflake. But if we treat our tools and technology as a way to address cells and make this technology compatible with trying a lot of different things on a lot of different cells, then we are trying to make the time and cost that you will sink into this so small that you will actually try it, right? And so if you're looking to do something in the ocean, then it should be easier for you to look for all the ocean organisms that are available and use one of them and be able to do that, than to take E. coli and spend 10 years trying to engineer and to do exactly what you need in the ocean. Currently, it's probably easier for you to grab E. coli and modify it, but really it should be easy for you to choose from a catalog of organisms that are available and are easily modifiable and say, what is my application? I want to do climate relevant application. And if I want to do climate change related bio, I know I have a place to look for all the organisms that I need. So really our goal is to look at this as a technology focused effort and make all of the organisms that we touch available in some level, in some way for people who want to try.
Yeah, it's clear that Cultivarium's mission is really going to help with the bioeconomy. We talked about open sourcing it, similar to how the internet was built, and now that the internet is our economy. And Biden released an executive order to spur the bioeconomy. One of the goals that they stated was that in 5 years, they would want to sequence the genomes of 1 million microbial species and understand the function of at least 80% of newly discovered genes. So what are your thoughts on that? And how does Cultivarium play a role here?
Yeah, so first of all, super happy about that, of course. Goes without saying. I think it is wonderful to have such a huge investment in biotechnology and in new organisms specifically. Cultivarium, of course, is thrilled to see that kind of push from Biden's administration. The thing that we think about when we hear that is, yes, it's great to sequence 1 million organisms. It is really hard to find the true function of 80% of the genes. So genome annotation is currently mostly being done by taking a genome, running it through a server, and search for homologies or search for relatedness to existing genomes, and then assign a function. That doesn't mean that every gene was tested for that function. It means that we think it belongs to that function because of relatedness and homology to other genomes. And really, functional genomics is the hard part here. Understanding all of these new microbes will require exactly what we do. It will require culture, it will require delivery of DNA, and it will require engineering, because you can't figure out the true function of a gene if you haven't tested it. We're getting very good at doing the computational analysis of what genes are and what we can find as alternative for a gene. But really, there is no escape from trying to culture and trying to actually manipulate organisms to know how they work.
You're saying that it is easy to find the information through this computer technology, but you really need to test it in the real world, in the physical world. So you might have all information you need online or through Cultivarium or some other databases, and it could give you a direction, but the only way to truly know is if you actually do it in real life. You can't just rely on computation and AI. It could help you get far and point you in the direction, but you won't know until you try.
Yeah, absolutely. You have to try with your own hands in the lab.
And I just wonder, Nili, do you have any idea when you do that kind of functional genomics and you're doing that kind of homology comparison, what's the accuracy of finding the function of genes based on homology with function in other organisms?
I don't know the number for that. Honestly, I don't know that we tested enough organisms that we only have the genome for to tell you if that annotation was really precise. Anyone who worked with a non-model organism will tell you that what you think is going to happen is not necessarily what's going to happen. And that's wonderful. And that's the point of working with non-model organisms. You need to do things like single gene knockouts. You need to actually grow them and see if they aggregate. You need to see if they like or don't like the conditions that you grow in. Some microbes would grow, but only on a substrate. And then your entire liquid fermentation is not relevant if they can't grow in liquid, only on agar. And so theoretically, they have all the genes to grow on your feedstock, but only on solid, not in liquid. So things like that, where you really have to see how it works, can be addressed with high throughput functional genomics. So the first collection of knockouts in E. coli, where you actually have a strain in which one gene at a time is deleted from E. coli, the KO collection, that's available. That's an amazing resource for the E. coli and generally for the microbiology community, because you can get any strain you need that has this one gene knocked out, so you know exactly what's missing and how the strain behaves. That it took years to make. And we don't actually need to repeat the 10-year effort of knocking every single gene in every single organism to do that. We have things today like CRISPR-I. CRISPR inhibition allows you to do a multiplex experiment where you have an inhibitor of every gene, so an mRNA that could inhibit every gene in one pool. So in a single experiment, you're generating all of the variants that are required to inhibit one gene at a time. And that's a pool. That's a pooled experiment, right? That's a faster way of doing it. That's a different way of thinking about how to do one gene knockout at a time. It's not the same exactly as knocking out and deleting each gene at a time, but do you need that? So there's ways of studying and poking an organism to get an idea of what it does in the lab with your own hands that I think are much faster and much more technology savvy than the traditional way we used to do microbiology.
And it's funny when you talk about poking an organism, I just think back to you deciding not to become a zoologist because you didn't want to have to dissect animals in the jungle in real life.
True.
Here you are dissecting trillions of organisms under the microscope.
True.
How dare you?
I know. But I feel like there's a little bit of a difference between opening up an E. coli and opening up a tiger.
Yes.
Not entirely the same. Yeah. It's a little less adventurous in that way. We're not in the jungle, but like our lab is a kind of a zoo, a kind of a jungle.
Yeah, I have learned a lot by working with you guys and have become more fascinated with all the microbes that are in our world and learning about all the different biological processes that some of these microbes have. So I am curious, are there biological processes that fascinate you, and how do you think that they can be applied? Because for me, I really like bioluminescence. I think that's so cool. Think about having glowing plants in my home, create a cozy experience. Like I've been seeing more bioluminescence, but what other biological processes are out there that fascinate you?
Yeah, I think some of them are very publicly known and obvious. And I think bioluminescence is the most known one, maybe, and the most visually impressive. I think some of the really interesting processes that we don't fully understand or can't fully use yet is the way cells handle extreme stress. And so bacteria that are resistant to incredible amount of radiation or desiccation or salt or extreme pH. So bacteria that are growing in very high salt concentrations, they have special ways of handling that amount of salt and still surviving. Our bodies can't do that. Most organisms can't do that. So I think one of the first programs we had that I think just speaks to most people when we say that is an extremophile cohort. We have a list of extremophiles from very weird environments that we're trying to see how you can use them in the lab while still maintaining their special superpowers. And I think that's the most obvious ones that people look at: Antarctica and geysers and deep soil, and think of how these organisms handle those. I think other things that you could look at is things like biomining, how microbes handle metals and resources that we might be able to use. Treating our waste, specifically human waste, specifically for climate change, is something that microbes can definitely help with. And I don't think we use them enough to do that. All kinds of waste can definitely be degraded with microbes. We just need to find the right ones and use them wisely. And then I think there's a lot of technologies that we will find by just looking at how they work. So GFP was found not in E. coli, not in Cerevisia, one of the most useful genes, right, that R&D everywhere in biotechnology is using. CRISPR-Cas was not found in a model organism. A lot of the systems that we adopt for our biotechnology needs were found by just observing other biological systems and how they work. So even though the original cell, the original organism was not used for one application, you can import useful genes into an organism that you do want to work with.
Well, most of you have heard of CRISPR, the gene editing tool. GFP stands for green fluorescent protein. You can use this glowing protein to mark genes and confirm expression or function through fluorescence imaging. CRISPR was discovered when studying the microbe Halophorax mediterranei, and GFP was discovered studying jellyfish. Both are non-model organisms.
The success of GFP and CRISPR-Cas opened up so many applications in biology, but it also, the success of that one gene import into other organisms made it less urgent maybe, or made other organisms overlooked. Because if you take one gene and you move it over into E. coli and it works, then you don't actually need the original organism that you needed, that you looked into, right? And so I think that by us looking at new organisms as a whole system and see how they function in their own environments or in the lab under conditions we can manage, will give you a lot more biology to discover and new systems than taking one gene and moving it over, which is something we can already do.
Yeah, I think it's just worth it to say for our listeners that just a reminder, no matter where you go on the planet, there is a microorganism living there. And when Nili talks about extremophiles, you talk about the ones that live in like the salt marshes. I think of the ones that live at the deepest trenches of the ocean under pressure that is so extreme that it would kill a human. And yet there are these microbial communities that thrive there. You mentioned geysers and hot pools. So no matter where you go on the planet, biology has evolved to live in those situations. And there's a lot for us to learn from that. Even just recently, I've learned of new wild type, meaning undomesticated yeast strains or species that produce oils that you can basically skim off the top of a fermentation tank. They are naturally produced by the yeast. Those are being used as biosurfactants. And then I mentioned to Henry about ectoins, I believe is what they're called.
Yeah.
Protein or a carbohydrate that certain bacteria produce under stress that apparently have tremendous cosmetic, beauty, personal care applications. So there's a lot to be learned from the natural world. And I think the tools that you guys are creating will enable a lot more of that discovery.
Yeah, absolutely. Halophiles, salt-living bacteria, a lot of them make actoins and related materials. And really, you shouldn't go as far as geysers and the pole to find things. You can just go to the New York City subway system. I am sure you will find very resistant microbes and rats.
Yeah.
Carrying their own microbes.
Exactly. Yeah. Carrying their own very
microbes and macrobes.
Resistant microbes.
Can that be a word? Macrobes from multicellular organisms? I don't know. Maybe we should coin that term.
We should.
Macrobes.
You should submit that to the committee of naming the new microbe. Half the microbes we have are extremely hard to pronounce. I can't say half the names properly because it's all Latin, very long names. They really should be more reasonably named.
We do have the superstar extremophile, which is the tardigrade that everyone I think knows. It's like the mainstream, cool extremophile. You guys are working with tardigrades, right?
We are working just with single cells.
Okay.
And so we are not currently working with tardigrades, but we are working with the equivalent microbial cells that could be resistant to a lot of the same stresses and tensions.
Nice. So I have another interesting vision type of question, is that if I were to give you a grant to extract microbes from any place on Earth, which microbe and where would it be?
I don't know which microbe. I think interesting places to look at. My country has some of the lowest place on Earth, the Dead Sea. It is already being investigated, of course, but I think the Dead Sea could have a lot of interesting microbes that we could look at. I think actually human environments, we were joking about the New York City subway, but I actually think human environments and human waste are some of the places we would probably find the most useful microbes if we just looked for them. The COVID-19 epidemics, a lot of the diagnostics was done by just following the sewage system. It's a way to see what's in the sewage system, to see how the population is doing by looking at the waste. And so I think if you look at the plastic waste we put in the oceans, if you look at the mountains of waste that we are producing, maybe by looking at what grows on there anyway, we find solutions to degrade most of these things, because most of things that are tossed from our houses get to nature and something will degrade them, right? So we know of some materials that are more easily or less easily degradable, but I think to find solutions, we can look in those places for straightforward microbes that might serve us really well if we only knew how to work with them in the lab.
Yeah. So you would go to the sewer.
Basically, I would go to I would go to the sewer, basically. I think the Dead Sea is a little more nice and beautiful than the sewer, but yet.
Yeah. You mentioned, so would there be a lot of halophiles in the Dead Sea?
Yeah.
You love
them.
them.
The Dead Sea is interesting. It's so salty that not a lot of things can grow there, but I think just looking at things that could handle that environment would be very interesting.
Cool. Interesting. You could go to the sewer near the Dead Sea so you can check off both boxes.
You would hope there's no sewer next to the Dead Sea, but yes.
We are coming up on the hour. We always ask if you have a favorite business or biotech book or movie or show that you would recommend to the audience, something that you really liked or enjoyed.
Having a toddler had taught me about a lot of children's science books that are out there. And we have a whole library of books at home that she loves. We just got those beautiful photographic albums of animals from like a National Geographic type, or some very impressive animal pictures, and just expose children to how science and animals and nature look like beyond the 5 farm animals that every single children's book show them, so that they know there's more than just cows and sheep and dogs.
And chickens and cats.
Exactly. So I just find that there's a lot of interesting children's science books that are really insightful and help show kids at a very early age that things are out there that are not what we see every day. And just keeping that mindset of, I know things are out there that I can't see every day or that I'm not familiar with. And that's not necessarily scary. For example, I don't get the insect ones. You know how they have like insect books for children? That is where I can't handle the insect world with her. But I think my recommendation would just be to try to open up young minds, whether it's toddlers, whether it's college kids. I didn't learn about any really weird bacteria when I was in college, because most of basic biology is being taught based on model organisms. So I think that just opening up to the understanding that a lot of different microbes and a lot of different animals are out there and we don't know them. I would love to see more of those.
2 things. One is that I think a lot of microbes are vilified because of germs. So people don't necessarily think about the benefits of a lot of these microbes. So there should probably be more media and books about the good microbes. There are products now, probiotics. People are starting to understand that. But also, though, for the children's books, I think that's really fascinating. Thanks for recommending it. I'm also, of course, a science nerd, and I have a toddler as well. One book that I'd recommend, just because you're talking about it, and for you, you might already have it. You know how there's a book called Goodnight Moon? There's one called Goodnight Lab. And it's like, good night, thermometer, good night, lab coat. Good night, professor who's yelling at me to publish my paper.
Maybe I would recommend that one to undergrads. Not, I don't know if toddlers, but yes.
And then there's like quantum physics for babies. And I mean, they use like a ball to talk about like mass and acceleration. So he just thinks of it as the ball book, but I think it's more just tongue in cheek. Although it's true. It is like science.
I think just being aware of things being different. So like the funniest thing that I can think of right now is I did my master's with E. coli. I used E. coli, and then I started doing my PhD, and it was a yeast focused lab. And so I used to clone all my plasmids in vitro by using E. coli and restriction enzymes and these things. But then when you get to a yeast lab, they tell you, oh, we just transform all the parts and the yeast puts them together. And you're like, what? No one told me that. I was doing the E. coli thing. And that's the most useful to model organisms. And the lab doing this, different labs doing that. And that's very common, that you just don't want to switch to a different model. You don't want to do something you are not already used to doing. So I just recommend opening up to other options. And hopefully what we try to do is make it easier to switch.
Absolutely. Nili, this was such a great interview. Thank you so much for taking the time to speak with us today. We're looking forward to seeing you very soon.
Yes. Thank you for hosting me. It's a lot of fun talking about it. And I think the only thing I want to say is we love to talk to people who are interested. And we would love to talk to any kind of organization, nonprofit, for-profit, any kind of endeavor trying to work with non-model organisms that we can help. Please talk to us. We're happy to talk. We're happy to help.
Fantastic. Thank you so much.
Thank you.
See you, Nili. Bye.
Yeah. See you soon.
So, Karl, what did you think about that interview?
I love Nili. She's one of my favorite people. She is incredibly smart, so driven. I admire her so much. I feel like we're so lucky to be a part of her team. And I think the scientists that work with her are so lucky to work with her. She has insights that are so deep. I think it's worth pointing out, one of the things about Cultivarium that makes them very interesting is that they're open access. And the goal is to make their tools and their databases available to the public or to people who would use them. The idea behind that is that the internet, as we know, which is basically everything these days, was built on open source tools. Most people don't realize that there is a whole other computer language called Linux, which is open source, and anybody can work on Linux, make modifications to it. The modifications better the program, then the community accepts it. One of the theses behind Cultivarium is that making tools for microbiology open source or giving people more access will, in fact, increase interest in the bioeconomy. It'll make it easier to work with all these non-model organisms. And as a result, we will all benefit from that. And I hope that people who listen to this enjoyed it as much as we did.
Yeah. I really appreciate your analogy on the open source, that the internet was built on open source tools. And this is similar to what Cultivarium is doing to build the bioeconomy, so the equivalent of the internet. One of the things that Nili mentioned and you reiterated during the interview was the concept of biosecurity, which also has an analogy to the internet. So maybe not security, but maybe regulation is what I'm thinking, because there isn't really much regulation to what you're building on top of the internet, right? So there's still a lot of issues with people using social media. How can government regulate that? There's some regulations for commerce that came up after the fact, after something negative happened. So borrowing from the development of the internet and the way that our economy works today, how can we get ahead of any biosecurity concerns, doing thought experiments, which scientists are good at, but unfortunately, they won't be able to capture everything because biology is way more complex than source code for creating software programs. So I think that's going to be very interesting.
I think we should try to get Megan Palmer on here again, because Megan's a deep thinker on this, and not just on biosecurity. We also have our friend Miriam El-Garbi, I always get her last name wrong, who is a biosecurity expert. But there are a number of people out there who are talking about the bioethics of working with life. And it's a deep subject that is something that we should all be fluent in and be able to have smart conversations about, because It's not going away. And since we are working with life on this planet, it is very important for people to understand what the implications are. And biosecurity is something that's very serious. We just lived through a pandemic. You start off by saying Craig Venter was convinced that COVID was a lab leak, accidental or intentional. That's still a problem. People need to understand what is biosecurity. And that is something that I know that the folks at Cultivarium are grappling with, because these technologies are very powerful and they can have real danger. We always talk about the benefits of them, but it's easy to go to the dark side if we want to.
Yeah, actually, there's another conference happening soon. It's the Bio Conference happening in Boston, which is more biopharma focused. But the keynote is about biosecurity and the rapid development of vaccines. The speakers are all from different government agencies like the Department of Defense. And that's how this biopharma conference is starting. And it's like, I think it's about 2 hours before any other conversation. So this is how important it is that they're having every single leader from pharmaceutical companies around the world at this conference. And the first topic they're going to be talking about is biosecurity and the rapid development of medicines and vaccines. When I saw that, I was like, oh, okay, yeah, this is getting very serious. And now with Craig Venter, I'm like, all right, A plus B equals C. I don't want to be a conspiracy theorist, but I just think hypothetically, like we're saying, just be prepared for any situation. We do need to make sure we have access to the tools, the right people are in place to make sure that we're safe and secure.
Yeah, it's just worth just saying a couple things. One is we need to find a couple of biosecurity people to come on and maybe make that a regular part of this show, because I think it's an important thing. And then for the doubters, or the people who have been maybe conspiracy theorists around vaccine, this was a conversation that I had at Simba Beta. What many people don't realize is that the vaccine was able to be developed as quickly as it was because of the tools that have been developed over the last 20 years. This stuff didn't come out of nowhere. Part of it was sequencing, part of it was developing chassis for vaccine development, and there was a very large multi-government effort to be prepared for the next pandemic, because historically our planet has a pandemic every 100 years. So we were preparing for it and we were able to develop a vaccine. And when I give a talk about the development of biotechnology, I point out that at the beginning of 2020, when the pandemic started, we had no vaccine for COVID. At the beginning of 2021, about a year later, we had 100 vaccines or more in the clinic being tested for safety and efficacy. And we were lucky that at the end of 2021, a vaccine was developed that was effective. We could have easily not had a vaccine at all, but had we not had 20 years of development before, I think we wouldn't have seen anything developed as quickly as possible. And today, the COVID vaccine has proven to be very effective and very safe, and is one of the most studied medicines ever developed.
Yeah. And then all of that rapid development, now that we know we can do it, we should do it, right? And this is to go back to Cultivarium and them working on creating a platform, open source tool sets to use non-model organisms. There's just so many things that we can make. And this is something you and I riff on all the time, because we're like, okay, if we were to get this information from Cultivarium, this information being the culturing, the DNA delivery, being able to engineer a weird microbe that can do, the most basic example for biologists, but the most exciting example for me is bioluminescence. I want to be able to engineer mid-sized plants so I can have ambient lighting in my home. Or obviously that's one example, but then when it comes to biomaterials or flavors, there's just so much that can be unlocked if we have the information, the tool sets and the data that Cultivarium's creating, or other biologists or companies. But I just think that there's a lot. I know you have some ideas. What have you imagined that could be made from using new non-model organisms?
So I'll say two things. And one is, I just think since we had the conversation with Savine a couple of episodes ago, talking about K18 hair, being able to dye hair without the use of harsh chemicals and changing the way your hair looks. You mentioned luminescent, having luminescent hair that maybe changes color. Those, I think, are things that are going to be very possible and made possible with the discoveries that someone like Cultivarium is going to make. And that's not even a wild application, like that's going to happen. A more wild application would be to engineer a plant like bamboo, which I probably mentioned on the pod since it's one of my obsessions, so that bamboo grows faster. It already grows very fast, but so that it becomes more of a carbon sink. And then you use laminated bamboo for construction materials. But I will say something that we haven't talked about that much, and I think we want to get into in the future, is one of the impacts of working with non-model organisms and thinking about them in general is that we can start to localize production. And localizing production is something that doesn't get mentioned enough, and like how transformational it will be to be able to produce everything that a city uses in that city instead of having to ship things from far away. That is going to make a huge impact on climate, on the planet, on manufacturing, on brands. And that is something that I'm very much looking forward to. And I think that the tools that Cultivarium are developing are going to make that possible. So I really am looking forward to having someone come on and talk about localization, because I think it's a really underestimated, overlooked development that we're going to see for sure in our lifetimes.
Absolutely. Absolutely. With that, I think that's the pod. We have been talking a bit because there's so much exciting things are going on now that we had the tsunami of biotech innovation hit us last week in Oakland.
Thank you for listening. We really appreciate our listeners, those of you who met up with us at Simba Beta. It was great to see you. If you like this podcast, please like us, share us, and leave a review. We are looking for more reviews because the reviews help us and we will get more listeners. And our goal is to sing the praises of biotech and sing the biotech gospel. For those of you who don't know what's going on in this space, we're very excited by it. Everybody's talking about AI. We spend a lot of time talking about AI, but we think that what we see happening in biotechnology is what's happening in the physical world, and is equally important. Thanks for listening, and we'll see you next week.
See you later. Bye.